Genetics and Genomics in Medicine, Chapter 8, Identifying Disease Genes and Genetic Susceptibility to Complex Disease. Chapter 8 Overview. In this chapter, we will consider the identification of disease genes and genetic susceptibility to complex diseases. The topics that we will cover include identification of genes in monogenic disorders, approaches to identifying susceptibility to complex genetic diseases, and the contribution of environmental and epigenetic factors. Module 1. Identifying genes in monogenic disorders. Historical perspectives. Monogenic disorders and the genes that cause them began to be identified in the 1970s and early 1980s. Often it was because the protein had been extensively studied initially. An example of this is hemophilia, which often is caused by abnormalities in the factor III gene. Factor III had been extensively studied in the pig model system. The protein had been purified to homogeneity, which enabled the isolation of the gene encoding pig factor III. Subsequently, the human homologue of pig factor III was isolated using the pig gene as a probe. In addition, cells which are highly enriched in a particular species of mRNA can also lend to the identification of the causative gene for a monogenic disorder. In the case of beta thalassemia and alpha thalassemia, the alpha and beta globin genes are produced in high amount or expressed in high amount in red blood cells. So the isolation of the mRNA from these genes was helpful in identifying the gene that was causative of this disease in individuals. In general, early approaches to identifying the genes that were causative of monogenic disorders involved what is referred to as positional strategies. That is, using a variety of approaches to narrow down the region of the chromosome or the loci where the particular gene of interest was harbored. In the case of X-linked disorders, this was somewhat easy from the perspective that individuals who have a disease which is X-linked primarily displays in males. And so immediately scientists could know that it seemed that these genes would be on the X chromosome, and that helped to set apart all the other chromosomes that we have in our cells. In addition to that, linkage analyses were performed where different types of experimental approaches would aid in identification of loci or regions of DNA that held the gene of interest. Hundreds of different markers are needed for this type of approach, and the technique of RFLP, or restriction fragment length polymorphism, helped to lead to the first map of genetic markers, which was produced in 1994. An important term to be familiar with in dealing with positional strategies is a haplotype. And a haplotype refers to a particular series of alleles at two or more neighboring loci on a single chromosome. And so those particular alleles will co-segregate with one another due to the fact that they are very rarely separated during the recombination that occurs during the meiosis phase of cell division. Let's take a look at an example of this. This is a mapping of the human meiotic crossovers that occur genome-wide using a select 3 SNPs that appear in families. And you can take a look at this, and the pink bands show the maternal grandmother, the blue bands show from the maternal grandfather, and the gray bands refer to areas that are heterochromatic and are not useful for this particular analysis. And you can clearly see in this diagram there are certain regions where the ability to segregate a particular SNP is limited. So there are some SNPs that persist despite many crossovers that occur and mix up the genome. So as we start off at the top, this example shows the deduced pattern of meiotic recombination in the chromosomes passed from mother to her daughter after whole genome SNP typing of the family members, including the grandparents. The egg transmitted by the mother to her daughter was formed by meiotic cell division, in which chromosomes from the maternal grandmother, shown in pink, recombined with those from the maternal grandfather, shown in blue, to give the alternating blue and pink patterns that are shown. As an illustration, the green arrows show the location of 3 crossovers on chromosome 1. More than 50 crossovers can be detected, but none are apparent, unexpectedly, on chromosomes 11 and 14. This slide shows inheritance of a disease haplotype in an autosomal dominant disorder. In A, the disease locus, highlighted by the yellow box, can be imagined to have 2 alleles, the D, or diseased allele, and the N, or the normal allele. Here, we also consider alleles at 4 marker loci that are physically located very close to the disease locus, for example, within 0.5 megabase. The disease haplotype in the affected individual is defined by the sequence of alleles at consecutive neighboring marker loci that could be represented here as 2-3-1-4. In B, the haplotypes in A belong to the effective grandfather 1-1 in this pedigree. The highlighted disease haplotype, dark blue A, is transmitted without change to affected individuals in generations 2 and 3. So this slide shows how the disease haplotype is spreading throughout 3 different generations. And you see they all move together as a group. So this is what haplotype is and why knowing haplotype is important if you're using positional strategies to identify specific loci. Something that's important to keep in mind when we're trying to track down disease genes and to positionally identify them is recombination. So remember, we have to look at that, and that's why linkage analysis really works, because during recombination, certain haplotypes segregate with each other. In this figure, each dot represents the number of recombinations identified in an individual meiosis. Each vertical line of dots represents the scores determined in each multiple meiosis in a single male or female, as shown. The number of recombinants per meiosis was determined by genotyping individuals in families with 6324 SNP markers. In an idealized situation, recombinants and non-recombinants can be clearly identified. In an autosomal dominant pedigree, an affected individual is heterozygous for disease allele, and he's also heterozygous for a marker. To get around the difficulties of identifying recombinants in human genetic mapping, sophisticated computer programs are needed. To do so, they do not attempt to make individual recombinants. Instead, their job is to survey the linkage data and then calculate alternative probabilities for linkage and non-linkage. They then express the ratio of these probabilities as a logarithmic value called a LOD score. And sometimes this is written capital LOD, and sometimes it's written lowercase LOD. An LOD score is the measure of the likelihood. It's a mathematical quantity. It's the measure of the likelihood of genetic linkage between loci. And when I say mathematical, I really mean it's a statistical, because it's a probability function. The log base 10 of the odds that the loci are linked with recombination fraction Q rather than unlinked. For Mendelian characteristics, an LOD score of greater than +3 provides minimal evidence and is the threshold value necessary to establish linkage. LOD scores can also be useful to exclude linkage, and the minimum LOD score for exclusion is -2.
Exome sequencing. Although many genes underlying monogenic disorders have been identified by the methods that we have described in this lecture, some monogenic disorders have not been well studied because they are very rare or because they are not readily identifiable. There may be difficulties, for example, in recognizing individual phenotypes within complex sets of overlapping phenotypes, such as intellectual disability. More than 7,000 monogenic disorders are estimated to exist, and although the methods have been very successful in gene identification, a new approach was needed to identify genes in the substantial proportion of monogenic disorders that are very rare or where there's difficulty in distinguishing the phenotype from related disorders. What is this new approach? That would be the massively parallel DNA sequencing, so-called next-gen sequencing. And we've talked about this earlier in this course in the methods that we have described in DNA sequencing in Chapter 3, and in the popular methods later we'll talk about in Chapter 13. This method offers a vast increase in the sequencing capacity and time taken to sequence a human genome. It's fallen from several years to just a few days, and soon just a few hours. The expense also has dropped dramatically. The inevitable consequence has been an explosion of genome sequencing. This slide shows the notion of exome, that is just the coding region, exome sequencing. So exome capture and sequencing. In this procedure, a genomic DNA sample, as shown in the workflow, is randomly sheared, and the fragments are used to construct a DNA library. Library fragments are flanked by adapter oligonucleotide sequences, which are not shown in this picture. The DNA library is then enriched for exon sequences, orange rectangles shown in this picture, by hybridization to DNA or RNA baits that have been designed to represent the human exon sequences, which are shown as red rectangles. The baits have a biotin group, depicted as a green circle, attached to their ends. After capturing exon sequences from the test sample by DNA hybridization, the biotin-loaded baits can be selected by binding to magnetic beads, which are shown in gray, coated with streptavidin, which are the yellow goblet shapes. And streptavidin actually has a very high affinity for binding to biotin, and the streptavidin-biotin exon complexes can be removed using the magnet. The captured exon sequences from the sample DNA are then subjected to massively parallel DNA sequencing, and the data are interpreted. And this slide just depicts that larger capture of those fragments using the biotinylated tags and the streptavidin coated beads. So it's a very straightforward workflow. After the DNA sequencing has taken place, then there is additional analytical procedures that have to take place that require the use of computational biology. There is mapping of the DNA, alignment to make this one contiguous sequence because obviously these are generating fragments, and then finally variant calling because you're going to align the sequence that you have from the genome that's been sequenced to a reference genome to determine whether there are any variants. In addition to identifying specific genes, pathways of known and novel genes that are involved in a particular process can be identified using next-gen sequencing approaches as well. This picture depicts known and novel genes for intellectual disability that are part of a protein and regulatory network. In a single exome sequencing study, 50 novel genes were identified for recessive intellectual disability. The novel genes were predicted to encode components of protein and regulatory networks that had been implicated by studies of known genes for intellectual disabilities, including the transcriptional translational network, the cell cycle related network, and the RAS-Rho PSD95 signaling network. Module 2, Mapping and Identifying Genetic Susceptibility to Complex Disease. It is convenient to divide genetic phenotypes into monogenic phenotypes influenced predominantly by a single genetic locus, and polygenic phenotypes. Multiple genetic factors determine the phenotype, each making small contributions. Polygenic diseases must be considered because monogenic disorders are quite rare. In addition, most genes have modifier loci, that they do not, any one gene does not operate in isolation and can be influenced by other loci near or distant from the gene of interest. And then in addition, there are also a variety of non-genetic factors such as epigenetic, environmental, and stochastic or random factors. Human traits can be divided into two classes. Some, like diseases, are dichotomous. You either have the trait or you do not. Others, such as height or blood pressure, are continuous or quantitative traits. Everybody has the trait, but to differing degrees. To explain quantitative traits, polygenic theory envisions the additive contributions of variable alleles from multiple loci. Many alleles at the underlying quantitative trait loci might have subtle differences, causing modest changes in expression levels of certain genes. Different combinations of alleles at multiple loci might then produce traits of continuous variable, such as adult height. This slide depicts a graphical representation of the distribution of a particular trait within the population, and again within first-degree relatives. And you can notice that there's a greater proportion or probability of individuals to have that trait if they are first-degree relatives relative to the general population. One can calculate and come up with a metric for this, and it's referred to as the risk ratio, or lambda. The risk ratio is the disease risk to a relative of an affected person divided by the disease risk to an unrelated person. Is designated as lambda with a subscript that defines the relationship. For example, lambda S represents the comparative disease risk for a SIB, brother or sister, of an affected individual. Unlike a monogenic disorder, for which the risk to family members are fairly precisely defined according to the simple theoretical calculations of the Punnett square and Mendelian inheritance, the risks for complex diseases quite often have been empiric. That is, they have often been based on surveys of incidence in families. The risk ratio lambda is a measure of how important genetic factors are in the etiology of disease. Not surprisingly, therefore, extremely high lambda S values were found in monogenic disorders. For example, in populations of Western European origin, the general lifetime risk of cystic fibrosis is about 1 in 2,000, but the risk to a fetus is 1 in 4 if parents have had a previously affected child. In that case, lambda S is 1 by 4 divided by 1 in 2,000 equals 500. For some complex diseases, lambda S values can be quite high. This table depicts the approximate relative risk ratios for SIBs compared with the general population for several selected complex diseases, averaged from many studies. So you can see, for example, if you have a sibling with Alzheimer's disease late onset, you're at higher risk. Crohn's disease, multiple sclerosis, and type 1 diabetes all carry a relatively high risk ratio, or lambda S. This data, as we stated, is derived from family studies, and it does, however, suffer from the negative that it neglects environmental factors. Twin studies often offer an insight into the relative influence of environmental factors, which is often ignored in simple family studies. An alternative to twin studies is also adoption studies, where siblings have been adopted out, and that helps to then factor in the factor of twins, or of an environment, I'm sorry. So type 1 diabetes, if you look at monozygous twins, has a lambda S of 42.9, whereas in dizygotic twins, it has a lambda of 7.4. So you can see there's a difference depending on which type of twin that you can have, whether it's a dizygotic twin or whether it's a monozygotic twin. So monozygotic twins, remember, are derived from the same egg, and dizygotic are derived from 2 individual eggs. The linkage analysis used to map genes for monogenic disorders are said to be parametric. That is, the data can be analyzed only if a specific genetic model is assumed. The data can be analyzed only if specific factors are present, and then details are needed for certain key parameters, such as the mode of inheritance, disease gene frequency, and penetrance of disease phenotypes. Parametric lineage analysis has been very successful in mapping the genes from Mendelian disorders, but they have been more limited in applicability in complex disease because of the general difficulty in providing all of the required parameters. For several complex diseases, there are also subsets in which the disorder shows a clear Mendelian inheritance. That is the most obvious when there is a dominant or quasi-dominant inheritance, such as in early onset forms of Alzheimer's disease, Parkinson's disease, diabetes, and various types of cancers. Large pedigrees, such as the one shown in this figure, offered an early way to identify genes associated with a complex disease phenotype. In early onset Alzheimer's disease, as shown in this figure, these approaches identified 3 disease genes, the amyloid precursor protein APP, and 2 genes involved in the processing of the APP protein, which we will discuss later in this lecture. In the identification of disease susceptibility genes, A significant candidate chromosome region can be identified, but it still remains a challenge to identify the specific gene within a locus. Despite these difficulties, genome-wide linkage studies have had a measure of success in mapping susceptibility genes in complex disease in specific candidate regions that would then allow subsequent gene identification using other approaches. This particular slide depicts the Crohn's disease susceptibility factor NOD2 and common variants and their expression in Paneth cells. So in A, the actual gene is shown and the different regions that were identified as being important in the function of the gene product or the protein that this gene produces. And then on the bottom is shown a immunohistochemistry image depicting where the expression is. So this has been very pivotal in identifying the actual role and developing treatments as well as diagnostics tests for Crohn's disease. Control studies can be carried out in which genetic variants are genotyped in affected individuals, which are the cases, and in controls, which are those that don't have the disease. Different methods can be used to measure the disease risk for each tested variant, notably the odds ratio, that is, the odds of being affected when processing a specific genetic variant, divided by the odds of being affected when lacking the genetic variant. This table shows a worked example of the odds ratios in a case-controlled study. This slide predicts examples of HLA-associated disease in Norwegian populations. All except one of the associations shown here shows an odds ratio greater than 1, which indicates disease risk. But in type 1 diabetes, HLA-DQ6 is protective allele, actually, and carriers have less disease risk than the general population. But you can see through looking at the odds ratios, there are several different diseases that are associated with HLA variations, and that has been picked up, and the odds ratios have been calculated to demonstrate this relationship. Shared ancestral chromosome segments and linkage disequilibrium in the immediate vicinity of an ancestral mutation. The more distant a chromosome ancestor is, the smaller each shared chromosome segment will be, but the larger will be the number of people who share it. Here in this image, we imagine a highly idealized situation where sharing of two chromosome segments on a single chromosome extends to all 8 individuals in a generation, generation 4, who have common great-grandparents. The extent of the shared region is greatest in the sibs, but progressively decreases the further the individuals are separated from a common ancestor. In B, linkage disequilibrium around an ancestral mutation that confers disease susceptibility. In the upper panel, imagine that the newly emergent mutation, indicated by a red asterisk, appeared on a chromosome that had a minor, infrequent allele at very closely linked SNP location, where allele 1 is the major allele. Affected individuals will have a significantly higher frequency of allele 2 at the marker locus than in the general population or than of control unaffected individuals. Haplotype blocks. Initial attempts to define ancestral chromosome segments began with high-resolution mapping of haplotype structure in defined small genomic regions and populations of European ancestry. The results suggested that our nuclear DNA might be composed of defined blocks of limited haplotype diversity, otherwise known as haplotype blocks. This figure illustrates an example of a haplotype block 84 kilobases long that spans most of the RAD50 gene at chromosome 5 q31. 8 common SNP loci were genotyped in this block, and 2 alleles at each of the 8 SNP loci means the potential for 2 to the 8 equals 256 different haplotypes. Yet, within this block, almost every chromosome 5 that is tested has one of only 2 of the 256 possible haplotypes, either the orange one shown in figure 1a, which we can abbreviate by listing the nucleotides at the 8 consecutive SNP loci as, or green, as the other haplotype. Overall, about 85% of our nuclear genome is mosaic structure composed of haplotype blocks. The average size of haplotype blocks in the population of European and Asiatic ancestry was 5.9 kilobases with an average of about 3.6 different haplotypes per block. How genome-wide association or GWAS studies are carried out. This slide depicts a typical GWAS workflow. Using haplomap data, representative SNPs are selected that will differentiate or tag the common haplotypes at each locus. In this example, 3 common haplotypes, A, B, and C, at locus 1 are tagged by 4 SNPs with strong color if present or gray if absent. But just 2 SNPs, purple and blue, are sufficient to discriminate between the 3 haplotypes. Similarly, the 2 haplotypes at locus 2 can be distinguished by either the red, chosen here, or the blue SNP. The tagged SNPs are then genotyped in disease cases and controls using microarrays, and the alleles frequencies for each SNP are compared between the 2 groups. SNPs associated with disease at an appropriate statistical threshold are genotyped, and in a second independent sample of cases and controls to establish which of the associations from the primary scan are robust. Visualizing and analyzing GWAS data is challenging and complex. Quantitative quaterile plots of the distribution of observed test statistics in a GWAS study are shown in this slide. In a case-controlled studies, a chi-squared comparison for absolute genotype counts is calculated for each variant. Red dots indicate the idealized test results, and blue dots represent the expected values under the null hypothesis of no association. In A, the test results are consistently inflated across the distribution, which could signify population stratification effects. In B, there's little evidence of an association from population substructure effects. Most of the red and blue dots coincide, but the deviations at the highly significant end of the scale in this example plot are compelling evidence for susceptibility with large effects. In C, a genome-wide Manhattan plot, think of skyscrapers, display genome-wide association findings according to their genomic positions on the horizontal scale and statistical significance on the vertical scale. The negative log P scale helps reveal signals of particular interest. This plot is from a large study of coronary artery disease that shows newly discovered disease susceptibility loci in blue and previously discovered ones in red. The dashed horizontal line at position 7.30 on the vertical scale indicates the threshold of statistical significance corresponding to p equals 5 times 10 to the negative 8 in this case. The most significant associations were with previously recorded SNPs in the immediate vicinity of closely neighboring CDKN2A and CDKN2B genes at chromosome 9p21. This is just a representation or blowout of the particular Manhattan plot for the previous genome-wide association study of a cardiovascular disease susceptibility loci. Genome-wide association studies in and of themselves can be quite limiting, but taken into consideration with other types of studies of a finer level, one can mine additional data. So, for example, the HLA association with rheumatoid arthritis is mostly due to 5 amino acids that are located in the peptide binding grooves of 3 HLA proteins. The study of these proteins and the identification of the specific consequences of these genetic variants help to further solidify the inklings that were obtained from genome-wide association studies. So you really have to take multiple approaches, not just the genome-wide association studies. Those can kind of provide clues, which then can be followed up using other methodologies and approaches. One concept is the common disease, common variant hypothesis. In this hypothesis, different combinations of common variants at multiple loci are believed to aggregate in specific individuals to increase disease risk. This hypothesis offers an explanation for why there's such a steep falling away of disease risk in relatives of probands with a common disease. It was supported by known associations of common variants with complex diseases, for example, in associations of common variants with Alzheimer's disease, as shown in this picture. The risk of late onset Alzheimer's disease for 3 human APOE alleles and amino acid differences of human alleles in the APOE of great apes. Compared with those people who are homozygous for APOE epsilon 3, the most common allele, people with 1 copy of the APOE4 allele have roughly a 3-fold greater risk of Alzheimer's disease, and people who have 2 APOE epsilon 4 copies have a roughly 15-fold risk. The mature protein made by APOE is called APOE with 3 common alleles, APOE2, APO3, and APOE4, each with 299 amino acids. They differ at positions 112 and 158 only, as shown. Chimps and gorillas, interestingly, have a non-polymorphic APO protein that resembles APOE4. Dots indicate amino acid substitutions between human APOE4 and the chimp or gorilla APOE. Black dots indicate 6 amino acid differences that are common to chimp and gorilla. Orange dots indicate 2 extra differences in chimp and gorilla and 1 in gorilla. The human APOE4 allele is regarded as our ancestral allele. Subsequent mutations gave rise to arginine and cysteine substitutions at positions 112 and 158 that increased the frequency presumed selective advantage conferred by these variants. The odds ratios of common risk alleles identified by genome-wide association studies are generally very low. Doubts about how much common variants could contribute to total disease susceptibility have propelled strong interest in an alternative explanation that rare variants originating by comparatively recent mutations account for much and perhaps most of the susceptibility to common complex diseases. One rationale for the common disease rare variant hypothesis is that given the multifactorial heterogeneity of individual single gene disorders, it's not very clear why complex diseases should be any different. At one extreme, many complex diseases are known to have Mendelian subsets in which the pathogenesis is due to very rare mutations of extremely strong effect, so that the phenotype is highly penetrant. As long ago as 2002, reported mutational studies of the NOD2 gene had indicated that there may be a heterogeneous set of rare NOD2 variants in addition to the 3 common alleles that were shown in Figure 11A of your textbook. And so it's not too much of a leap to propose that DNA variants associated with common disease might be heterogeneous, and different frequency classes might be envisioned with effects that are very roughly inversely proportional to their frequency. This figure gives a cartoon schematic demonstration of how different classes of DNA variants can contribute to complex disease. Common and moderate common DNA variants have reached high to moderate frequencies in the population because each originated by mutation a long time ago and have been passed down through very many generations to large numbers of descendants. The vast majority of the common or moderately common DNA variants are associated with disease of very weak effect. A very few have substantial effects, such as many HLA alleles in autoimmune disorders, and are represented by the cloud effect in the main boxes. Rare variants have originated fairly recently and so can have rather stronger effect than common variants, but they will eventually be removed by natural selection if the effects are strong enough. De novo variants can have large effects and are quickly removed from the population. Many complex diseases have Mendelian subsets in which the disease phenotype is caused by mutations that have a very strong effect, resulting in highly penetrant phenotypes. As described earlier in this course, copy number variation in the human genome can range from changes to single nucleotides to change in copy number of the whole chromosome. So the term copy number variant is poorly defined, but it can mean changes from the copy number of sequences of intermediate length, say just 100 nucleotides long, upwards to a few megabases. So although the number of copy number variants of loci is much less than the number of SNP loci, the larger size of CNV loci means that if we compare two genomes, the base pair differences between the copy number variants significantly outnumber those due to SNPs. And here we have some diseases that are associated with copy number polymorphisms. Those include Crohn's disease, coronary artery disease, psoriasis, and systemic lupus. In addition, there are examples of loci that frequently undergo copy number variation, such as those that are involved in autism spectrum disorder. So there are quite a few loci on a wide variety of chromosomes that undergo frequent copy number variation, and that is associated with autism spectrum disorder. Now, there are a number of limitations of genome-wide association studies, and that can be that large numbers of common variants have weak effects, so sometimes the associations can be mixed. In addition, rare variants that have large effects can weigh in on the ability of genome-wide association studies to make an accurate call. And genome-wide association studies definitely ignore any type of gene-to-gene or gene-environment interactions. There is no way for genome-wide association studies to account or to take into account those types of interactions. And in addition, genome-wide association studies, just by virtue of the methodology, cannot take into account any type of de novo sequence variants. It is interesting to ponder that after a long period of slow growth, humankind or the human population recently exploded. And because of the explosive population growth in the past few centuries, much of our genetic variation is relatively recent, and the great majority of individual DNA sequence variants that affect how our genes are expressed occur at relatively low frequency in the population. Despite the difficulties associated with making a direct association of rare variants to disease, there have been some great advancements made, and age-related macular degeneration is one example of these. Using genome-wide association studies, this complex disease had several genes, the CF8, CF1, C3, and C9 genes were identified as being causative in collection, in aggregate, to the AMD phenotype. Module 3, Environmental and Epigenetic Factors. Our developing knowledge of genetic architecture and complex disease and the contributions of environmental factors and epigenetic factors have contributed to a better understanding of disease. The success of and utility of genome-wide association studies is clearly exemplified by inflammatory bowel disease, including Crohn's and ulcerative colitis. We have been able to make inroads on the assessment and prediction of disease, as well as developing genetic tests. And recall that genetic tests really must be sensitive and specific. Sensitive in that they must detect all individuals that have the disease and therefore not lead to false negative results, and they must be specific in that people that do not have the disease do not test positive or lack of false positive results. And we really have been able to do this with Crohn's and ulcerative colitis, and this has not only led to genetic tests for these diseases, but also to novel therapies as well. This slide depicts genome-wide association success stories, and one can take a look, and clearly the numbers were low before 2007, and then with the advent of next generation sequencing technologies, additional variants were identified that have led to great success both in inflammatory bowel disease as well as autoimmune disorders. Predictive accuracy of testing for multiple genetic susceptibility factors in complex disease. A receiver operating characteristic, or ROC curve, plot sensitivity against specificity for a test. The figure shows ROC curve simulations with 2, 3, 4, or 5 independent disease susceptibility factors. In this case, susceptibility factors 1 to 5 are imagined to have relative disease risks of respectively 1.5, 2, 2.5, 3, and 3.5. For example, this would mean if someone has a relative risk of 2, that the person with a susceptibility factor has twice the risk of developing the disease compared to a person without it. Testing for multiple susceptibility factors can lead to an increase in the area under the curve, and the greater the area under the curve value, the more discriminating the test. So a ROC curve plots sensitivity against specificity. And here you can see the ROC curves for the different numbers of loci that are associated with the disease. And you can see that when there's 5 loci, for example, that are associated with this, if there's 5 susceptibility factors that are associated with a given disease, then the area under the curve is greater, and it seems to increase with the number of loci that one can have that are associated with a particular disease. This slide depicts one biomarker that was identified through genome-wide association studies as increasing susceptibility to Alzheimer's disease. The gene that encodes gamma secretase, a protein that plays a role in the buildup of amyloid beta in the brain, was associated with increased risk of Alzheimer's disease. And that is because simply if there are genetic variations in the gamma secretase, then it does not perform its function properly, and that in turn leads to an accumulation of the amyloid beta, leading to the formation of amyloid plaques. Another key feature is that early onset Alzheimer's disease genes and susceptibility factors for late Alzheimer's belong to common pathways, which in turn identify targets for potential therapeutic agents. The gene symbols highlighted in blue in the top left are early onset disease genes identified by parametric linkage analyses in which causal variants are highly penetrant. Loci implicated from genome-wide association studies are highlighted in yellow as common disease susceptibility factors with generally modest or weak effects. They include the previously implicated APOE gene, which was moderately strong effects and is shown with a distinguishing red border, plus some other well-replicated genes. And this has led to a deeper understanding of the large pathway that leads to the accumulation of neurofibrillary tangles, senile plaques, and ultimately neuronal death. Genome-wide association studies provide valuable insights into the pathogenesis of inflammatory bowel disease. Maintaining intestinal homeostasis involves a coordinated action by epithelial cells, which include the goblet and the Paneth cells, and cells from both the innate and adaptive immune systems. Barrier cells permeability permits microbial incursion that is detected by the cells of the innate immune system. Innate lymphoid cells orchestrate appropriate responses, in part by releasing extracellular mediators that recruit cells of the adaptive immune system, including B cells, helper T cells, and regulatory T cells. Genetic variants, the microbiota, and the immune factors affect the balance of these signals. Genes in linkage disequilibrium with IBD-associated SNPs are classified according to their functions in the context of intestinal homeostasis and immunity. Numbers of genes in each biological category are shown in brackets, but the understanding of these genes and these pathways can lead to improved disease prevention as well as treatment. Mining the GWAS to create heat maps. Heat maps that show correlations between different complex diseases according to genome-wide association studies are shown in this figure. Positive relationships between pairs of diseases are shown in the olive green colors. Negative relationships are shown in purple colors. The abbreviations on the vertical and horizontal axes denote diseases with an autoimmune component, shown in magenta, inflammatory disorders, orange, or other diseases in black. Hierarchical clustering using these correlations as a distance metric is shown on the left. Figures in red at the left are approximately unbiased probability values for each cluster, indicating how strongly the cluster is supported by data. Diseases with high probability values larger than 95% are strongly supported. Investigations are capable of identifying factors that confer protective against disease risk. In this table, examples of protective factors that are identified in various common diseases are shown. And you can see that there's a wide variety of protective factors against diseases such as Alzheimer's disease, coronary artery disease, Crohn's disease, rheumatoid arthritis, as well as genetic variants against infectious diseases such as malaria, HIV/AIDS, and norovirus. And a good example of how the environment interacts with genes. So, one of the focuses of this particular module is gene-environment interactions. So here is a slide that depicts the possible implications of interactions between the FUT2 non-secretory status and the gut microbiome in the pathogenesis of type 1 diabetes. The common FUT2 non-secretor allele alters the profile of mucosal glycosylation and so prevents microorganisms from adhering to the mucosal epithelial cells and the mucus layer lining the gastric epithelium. This null allele was naturally selected under evolutionary pressure to protect the host against bacterial, fungal, and viral infections, but it also imbalances the microbiota of the gut. A possible consequence is decreased antigenic stimulation to the immune system early in life, which is, common in modern societies, that we would predispose individuals homozygous for the non-secretor allele to an increased risk of developing type 1 diabetes. So it's an interesting thing to ponder, that your gut microbiome might in turn predispose you to diabetes, but your genetic particular genotype would dictate to what extent that would be the case. Other examples of different types of environmental factors that contribute to common non-infectious and non-cancerous disease are shown in this table. They can include things such as tetarogens and abnormal metabolite levels in uterine environment, and they can lead to neurotube defects such as spina bifida, and unbalanced diets such as the overconsumption and excess fatty foods that's seen in type 2 diabetes, smoking, which leads to increased risk for many disorders such as coronary artery disease, age-related macular degeneration, Crohn's disease. So it's interesting because we don't really know. We know that there are genes that are involved, and we know that people that smoke then we know that the genes are involved, so the individuals that have the disorder have those genetic variants, but then we also know that individuals that, say, smoke, for example, have a greater risk or a more severe phenotype of AMD or cardiovascular disease. And so in some way, that environment is perturbing the gene in some way, or exacerbating the phenotype. And this is also exposure to infectious microorganisms, peptic ulcers that are caused by H. pylori infection, or commensal microorganisms. And that was the example that we were talking about earlier with the gut microbiome. And here we show the importance of gene-environment interactions using HDLC as an example. According to different total levels of dietary fat intake, variations in the predicted values of high-density lipoprotein cholesterol are shown for 3 different genotypes of a particular polymorphism at a particular lipase locus. And so you can see that there is a distinct interaction between the environment and the different loci for these genes. And not surprisingly, there is, when we look at our nuclear genome, of course, there are variations, but also in the mitochondrial DNA, there are various haplotypes. As mitochondrial DNA is strictly maternally inherited, the mitochondrial DNA undergoes negligible recombination at the population level, and so SNPs in the mitochondrial form branches an evolving phylogenetic tree. The major subdivisions of the world mitochondrial phylogeny occurred more than 10,000 years ago and are called the mitochondrial DNA haplogroups. And they developed in humans as they migrated to new geographic regions, leading to region-specific haplotype variation, as shown in this slide. One way that scientists can understand, or geneticists can help to understand, the effect of environment on genes is by carrying out prospective cohort studies. And so this is a table that shows the example of the UK Biobank prospective cohort study, and they collected DNA samples from a wide range of individuals, but in addition to that, they conducted baseline questionnaires, baseline physical measurements, and follow-up and future measures to see if there's a particular, if you look at a group of individuals over longitudinal time, to see how the particular genotype varies with their environmental and behavioral phenotypes. And last but certainly not least are the influence of epigenetics on our susceptibility to disease. And there are a wide range of experimental investigations that are underway to better understand the global patterns of DNA methylation, as well as nucleosome organization. We look at epigenetics as things such as and it's very dynamic compared to the stability of our genome. Effectively, the chromatin states across all chromosomes are dynamic, and they're determined in terms of the patterns of cytosine methylation, histone modification, and the position of nucleosomes. And that's very fluid and responds to different environmental cues, and in turn that can influence whatever genetic variation there may be in the individual. In addition, the epigenetic changes that occur over time in aging. So we may have both genetic and epigenetic changes that occur over time and increase or change and can lead to the things that we associate with aging, such as a decreased ability to respond to DNA repair. So there may be a factor, an epigenetic factor in aging as well, and can then in turn influence our susceptibility to genetic diseases. We also think that perhaps epigenetics may be what influences changes that are observed in monozygotic twins. Monozygotic or identical twins are extremely initially similar in appearance and behavior, and then they go on to develop significant differences in the various aspects of what is called a healthy phenotype. So not all from a genetic perspective, they may be similar, but because of epigenetics, the actual way that that a particular genotype manifests itself can be different in what would essentially be the same in a monozygotic twin. So all of these are very interesting questions that are currently subjects of active research. And finally, there are many unanswered questions in the area of transgenerational epigenetic effects. And some of these we are being able to understand not just by looking at human model systems, but by looking at things such as model systems in C. elegans and even in plants. So things such as availability of food, our epigenome may be influenced several generations back and is still manifesting itself in ourselves, in our generations today. So this concludes the chapter 8, and thank you, and I hope that you've learned a lot.